Dear students, the topic is transduction in bacteria. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Transfer of DNA or gene from one bacterium to another through a bacteriophage or virus. Bacteriophages are the virus which attack bacteria. Transduction was discovered by Zinder and Lederberg in Salmonella. Hershey and Chase used transduction as a tool to study or to prove DNA as the genetic material. In this process, when a bacteriophage infects a bacterium that utilizes the bacterial genome for its multiplication, during the process of multiplication of viral genome along with the bacterial genome, sometime viral genome will be packed up with the bacterial genome, and when lysis occurs, the viral genome will carry some part of the DNA or bacterial genome along with it. When these viruses attack another bacterium, which release viral genome and bacterial genome into the host cell. So that time the viral genome utilizes only the bacterial genome for its multiplication process. So what will happen to this bacterial genome which is obtained from this bacterial cell? That will be remain as plasmid. That will be remain as plasmid in the new bacterial cell. If that bacterial DNA is homologous to this bacterial DNA, which will integrate and be joined with the same bacterial DNA. Maximum this bacterial DNA will act. This bacterial DNA will become plasmid in the bacterial cell. In transduction, there is no direct contact between two bacterial cells. Here, virus is the mediator to transfer a portion of DNA from one bacterial cell to another bacterial cell. When a virus carries this bacterial DNA into another one, which will remain as plasmid in the bacterial cell. Here is animation for transduction. A bacteriophage which infect bacteria use host cell to multiplicate, and while assembling they sometimes pack the bacterial DNA with them. Later, when these viruses infect new bacterial cells, the bacterial genome that they carry may get inserted into the host genome. The bacterial DNA either form plasmid or get inserted into the recipient DNA if it is homologous to the recipient genome. Most of the time it remains as an extra chromosomal DNA. It can also get inserted with the prophage if it is a temperate phage. So the fate depends on the portion of bacterial DNA and also on the nature of bacteriophages. Of transaction. One is generalized transaction and the other one is specialized transaction. In generalized transaction, any part of DNA can be taken by the virus from bacterial cell. But in specialized transaction, only a special part of DNA which will carry by the bacteriophage from the bacterial DNA. Generalized transduction. Generalized transduction can occur by both lytic or lysogenic cycle. Here, any random part of DNA gets packed in bacteriophages by mistake along with the viral genome. It occurs at the lytic stage of the phage life cycle. When the virus containing bacterial DNA infects another cell, it can get inserted into the host genome, or if it was a plasmid, then it can reform the plasmid. Generalized transduction is used to study linkage information, gene mapping, comparing genome of two different bacteria, mutagenesis, etc. Example of generalized transduction include E. coli transduction by P1 phage. The second type of transduction is specialized transduction. It can occur only through the lysogenic cycle, that is by temperate phage. Here, only the specific part of the bacterial DNA is packed into the virus. It occurs when the prophage, that is viral DNA which get inserted into the bacterial genome in the lysogenic cycle excises. When prophage excises from bacterial DNA, some parts of bacterial DNA which are flanked on both sides of the prophage are also excised. Here, the newly packed phage genome consists of both bacterial and viral genome. Later, when the virus with the recombinant genome infect a new bacterial cell, the bacterial gene also gets inserted into the host genome with the viral genome through lysogeny. The recipient cell now shows the newly acquired characteristics. Specialized transaction is commonly used for isolation and insertion of genes of choice. Example of specialized transaction include E. coli transduction by lambda phage. Application of transduction. Transduction is one of the most important tools for genetic engineering. It is used to insert the gene of choices in animal and plant cell to modify the genetic constituents and get the desired characteristics. It can be used for gene therapy. It's a huge potential to cure genetic diseases. It's an important tool in genetics and molecular biology research. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.